Welcome back to the deep dive. Today we're looking at a really fascinating protein, TRIM28.
That's right.
Maybe not a household name, but the sources you've gathered show it punches way above its weight in how our cells work.
Absolutely. It's part of this big TRIM family of proteins. And what's so interesting and maybe a bit tricky is just how many different jobs it has.
Right. It seems to touch everything from gene expression to cell survival.
Exactly. But the details, the specific ways it works, especially in disease,
Yeah.
that's still being mapped out as the sources make clear.
And that's where it gets really complex, isn't it? Because the research seems to point in, well, opposite directions sometimes, especially with cancer.
That's the crux of it. Sometimes it looks like it's helping tumors grow, other times like it's holding them back.
Okay. So our mission today is to sort of unpack all this information you've pulled together, the research, the articles.
Yeah, distill it down. What is TRIM28? What's its basic job? And crucially, what's the story with its complicated role in cancer?
Think of it as your shortcut to getting up to speed on this really key biological player.
Let's do it.
Okay, so first things first, let's break down the protein itself. What's it made of? What are the functional parts?
All right, so TRIM28 has several distinct sections or domains. At one end, the N-terminus, you've got the typical TRIM setup, a ring finger domain, two B boxes, and a coiled coil region.
That's the signature for the whole TRIM family, right?
Pretty much, yes, that core structure. Then moving along, there's this sequence called PXVXL and another called the TF1 signature sequence, or TSS.
Okay.
And at the other end, the C-terminus, there's a plant homeodomain, a PhD finger, and a bromodomain.
Wow, that's quite a toolkit. How do all those pieces actually function? What do they do?
Well, the R-ing domain is really important. It gives TRIM28E3 ubiquitin ligase activity.
Meaning it can tag other proteins with ubiquitin.
Exactly, which often marks them for degradation, like cellular garbage disposal. Then ubiquitination can do other things too, modify function.
Okay, so it's like a label maker for proteins?
Sort of, yeah. Then the main TRIM part, the N-terminal region, is key for interacting with a huge group of transcription factors called CARBAB-ZNFs.
CARAB-ZNFs, and that interaction is central.
Very central to its main job. And then the domains at the other end, the PhD and bromodomain, along with that PXVXL bit, they're crucial for recruiting other partners.
Like what?
Things like the NERD complex, which remodels chromatin, and an enzyme called STDB1, which adds silencing marks to histones. They also grab onto a protein called HP1.
And all that recruitment does what exactly?
It essentially helps pack the DNA tighter, create condensed chromatin. And tightly packed DNA usually means the genes in that area get switched off, silenced.
Ah, okay. So fundamentally, TRIM28 acts mainly as a gene silencer, a transcriptional core pressor.
It's its best-known role, yes. It partners with those CARBAB-ZNFs, brings in the silencing machinery, and puts the brakes on gene expression.
And is TRIM28 itself regulated? Can it be switched on or off?
Oh, definitely. Multiple layers of control. This production can be ramped up by proteins like WDR4 or shut down by others like ZBRK1. And after it's made, it gets modified too. Things like accumulation, ubiquitination. These can change its activity, where it goes, who it talks to. It could even modify itself.
It's amazing how intricate the control is. Okay, let's dive into that really confusing part. The sources highlight its dual role in cancer. It's not just one thing.
Definitely not. The evidence is pretty clear that it can be, well, two-faced, depending on the cancer type, the stage, what's going on in the cell. It can look like an oncogene or a tumor suppressor.
Okay, let's take the oncogene side first. Where does it seem to promote cancer?
Well, we see higher levels of TRIM28 reported in quite a few cancers: gastric, lung, cervical, breast, prostate. Quite a list.
And high levels are bad news.
In some of those cancers, yes. Higher TRIM28 correlates with poorer prognosis, suggesting it's driving the disease.
How? What mechanisms are mentioned in the sources?
Several key things. One is promoting the cell cycle, making cancer cells divide more. It works with MDM2 to shut down P53.
P53, the big tumor suppressor. So it helps cells ignore stop signals.
Exactly. And it also helps get rid of P27, another cell cycle break, by working with UBE2S. That's been seen in liver cancer, hepatocellular carcinoma.
Okay, faster division, less cell death. What about spreading, metastasis?
It's heavily implicated there, too, in driving EMT epithelial to mesenchymal transition.
Right, where cells become mobile and invasive.
Yeah, a critical step for tumor spreading. TRIM28 seems to regulate key EMT proteins like TWIST1, and it's linked to other molecules involved, like SNHG8.
That's huge for aggressive cancers. Anything else? Cancer stem cells?
Yes, another really important role highlighted in the sources. It seems crucial for keeping stem cells in their undifferentiated state. It can even block efforts to reprogram cells.
So it maintains that pool of self-renewing, hard-to-kill cells?
Seems like it. It's noted in breast cancer stem cells, working with EZH2 and SWI-SNF complexes to help form those tumor spheres you see in labs. And it affects markers like CD133, linked to stemness in several cancers.
Wow. Sustaining the root of the problem, in a way. Any other pro-tumor actions?
Yes. Promoting autophagy has mentioned that recycling process in glioblastoma, which can help cancer cells survive stress, and also promoting the Warburg effect, that metabolic shift cancer cells use, and liver cancer progression by degrading regulators like FBP1.
Okay, that's a lot of ways it can help tumors, but you said there's a flip side. Where does it act like a suppressor?
This is where context is everything. The sources show findings that really contrast with that picture. For example, in early-stage lung cancer, higher TRIM28 is actually linked to better survival.
Better. So the opposite effect.
Exactly. Suggesting maybe it's slowing down proliferation in that specific situation, perhaps by putting the brakes on E2F proteins, which drive cell division.
So early lung cancer, it might be protective.
The data suggests that possibility. And there's also evidence from mice. If you remove TRIM28 just in the liver, they tend to develop benign liver tumors, adenomas.
Suggesting it normally prevents those from forming.
Right, indicating a protective role against tumor initiation there. And it's also generally noted as important for keeping normal, non-tumor cells behaving properly.
So it's essential for normal cell stability.
Yes, which just reinforces that you can't paint it with one brush. Cancer type, stage, the specific cellular environment, it all matters for whether TRIM28 is friend or foe.
That complexity is, well, it's challenging, but also makes it incredibly interesting. Okay, let's shift gears slightly to another key area, cell death. We touched on apoptosis suppression, but the sources get into detail about a role in necroptosis.
Right. Cell death is huge in cancer, obviously, because cancer cells are masters at avoiding it. TRIM28 can block apoptosis, the quiet program death, through MDM2P53 and other interactions, but this necroptosis role is newer and quite detailed in the sources.
So first, remind us, what is necroptosis and why is it relevant here?
Necroptosis is another form of programmed cell death, but unlike apoptosis, it's often messy and inflammatory. It rings alarm bells.
Inflammatory.
Oh.
So that could be useful for getting the immune system involved.
Precisely. There's a lot of interest in triggering necroptosis in tumors to potentially wake up the immune response, make those cold tumors hot.
Okay, and how does TRIM28 fit into that process?
According to the sources, here's a key mechanism. During necroptosis, a protein called RIPK3 gets activated. It's a core part of the necroptosis machinery, the necrosome.
Right.
Activated RIPK3 then directly adds a phosphate group, phosphorylates TRIM28 at a very specific spot.
Right.
Serine 473 or S473.
Phosphorylation, the classic molecular switch. What happens when TRIM28 gets phosphorylated at S473?
This is the crucial part. That S473 phosphorylation stops TRIM28 from binding to chromatin to the DNA.
So it kicks it off the DNA.
Essentially, yes.
Yeah.
And since TRIM28's main job on chromatin is to repress genes.
If it's kicked off, those genes get turned on.
Exactly. It inactivates TRIM28's silencing function. So the genes it was keeping quiet are suddenly derepressed, activated.
And what kind of genes are they?
Inflammatory cytokines, things like IL-8, IL-1, TNF-alpha, CCL4, potent signaling molecules that draw in and activate immune cells. So necroptosis leads to this burst of new cytokine production.
Wow. So the cell death process itself uses TRIM28 as a lever to trigger inflammation by removing its repressive brake.
That's the mechanism laid out. It involves pathways like NF-κ, the master inflammation regulator, but also other factors like SOX9. And experiments confirmed this. A mutant TRIM28 mimicking phosphorylation can't repress these cytokines properly.
And this S473 phosphorylation is specific to necroptosis, not just any cell stress.
The Solon has emphasized that, yes.
Right.
They distinguish it from, say, DNA damage-induced phosphorylation, which happens at a different site, S824. This S473 event is shown to depend specifically on RIPK3's activity, linking it directly to necroptosis.
That's a really neat molecular link. Okay, how does this connect to the bigger picture of immune response and maybe even therapies?
It connects very directly. Those inflammatory cytokines released when TRIM28 is inactivated during necroptosis. They're signals. They can recruit and mature dendritic cells, DCs.
Which are key for kicking off an adaptive immune response.
Exactly. DCs present antigens and activate T cells, including the CD8 plus T cells that can kill tumor cells. So by silencing TRIM28's silencing effect, necroptosis might help rally the immune troops against the tumor.
Making the tumor environment less immune suppressive, more immune active.
That's the idea. And the sources back this up with data from TCGA, the Cancer Genome Atlas. They found that RIPK3 levels tend to correlate positively with immune cells like CD8 plus T cells and DCs being in the tumor.
Okay, more RIPK3, more immune cells. What about TRIM28?
That's the key contrast. TRIM28 levels correlated negatively with those same immune cells.
Meaning more TRIM28, fewer killer T cells and DCs around?
Precisely. Which strongly supports the model that TRIM28 normally helps suppress that anti-tumor immune environment. The sources also briefly mention other reports linking TRIM28 to keeping viruses latent and generally dampening anti-tumor immunity, though maybe the exact mechanisms there need more exploring.
Okay, so considering everything cell cycle, EMT, stem cells, this intricate role in necroptosis and immunity, TRIM28 seems like a really central node. Does that make it a drug target?
It definitely makes it an attractive one, yes. Because it's involved in so many pathways critical for cancer, interfering with it could have broad effects.
So people are thinking about drugs to block TRIM28.
For sure. Developing small molecule inhibitors that specifically target TRIM28 is definitely on the table, potentially as part of combination therapies.
Are there links to other existing therapies or pathways being targeted?
Yes, it seems interconnected. The sources mentioned, for instance, that inhibiting mTORC1 with drugs like retiferolimus affects TRIM28 phosphorylation in bladder cancer, and another drug, verteporfin, inhibits the immune checkpoint PD-L1 partly through a pathway involving TRIM28.
So it's already popping up in relation to other key cancer pathways and targets.
Right, it shows how deeply embedded it is in the cancer cell's network.
Well, this has been quite the journey into the world of TRIM28. To recap, it's this incredibly versatile protein, a master regulator involved in chromatin, gene expression, DNA repair, cell division, EMT, stem cells.
And complex roles in different types of cell death, like necroptosis and shaping the immune response.
And the big takeaway seems to be this perplexing dual nature in cancer, sometimes pushing it forward, sometimes holding it back, all depending on context.
Absolutely, you can't just label it good or bad.
Understanding these kinds of context-dependent players is just so critical, isn't it, for figuring out smarter ways to treat cancer and other diseases.
It really highlights how complex biology is and how tweaking one component can have so many downstream effects.
So here's a final thought to leave you with. We've seen how necroptosis can potentially boost anti-tumor immunity, partly by shutting down TRIM28 suppressive function via that specific S473 phosphorylation.
Right, lifting the brakes.
So could we potentially find ways to selectively trigger that specific inactivation of TRIM28 in tumor cells, perhaps alongside immunotherapy to get the immune benefits without necessarily enabling its pro-tumor roles elsewhere? It really underscores the challenge, but also the potential reward of learning how to precisely manipulate such a complex regulator like TRIM28 in our fight against cancer.